All right, so now we have to think about, I always start microbiology classes off with evolution topics, and because microbes were here first, and they were the first organism to come into existence on Earth. So I wanted to talk a little bit more about that. And that's another microbial art. Can you tell what that is? Does that look like a beer stein with beer? The bubbles and everything. I thought that was pretty cool that they were able to capture the bubble growth like that too. I don't know how they did that exactly, but that's pretty cool. All right, so let me go into, well, let's start there. Early life. So if finding fossils of microbes is pretty challenging. I would not want to be tasked with that myself. I think that would be incredibly challenging, but many scientists, evolutionary microbiologists do this. And really your best bet of finding fossil evidence of microbes is to go to stromatolites. So I want you to know that term. Stromatolites are layers upon layers of microbes that form mats. And then just over the years, they just, more layers keep accumulating until you can see it easily enough. So this bottom right picture that you're looking at here, that's actually like in the shallows of the ocean. I think this is off the coast of Australia. And you can walk amongst those. And so they can vary in size to, small to fairly large. And they just kind of look like weird rocks, but really they're layers of microbes. And most of the microbes in there are cyanobacteria. Do I have that? Yeah. So in the notes section, by the way, my PowerPoints, in the notes section, I often include additional information. So you might want to check out my notes area too of the PowerPoint slide sets. But cyanobacteria tend to dominate in these stromatolites. But the lower left image, you can see that layering idea. And then some, depending on the age of the stromatolite, they might be totally fossilized or they could still be living. So there's like living stromatolites out there too. It's not just all fossils. And so it just depends on the how long they've been in existence. But when they look at the fossil evidence of stromatolites, they start to think about, 3.4 billion years ago, roughly, was when we can see that this started happening. And so that's a long time ago. It's kind of hard to wrap your head around, but basically it is, what are they searching for? Fossils, that can be very daunting. Sometimes they look for biosignatures. That's the fancy word for something as a result of some biological activity, some kind of chemical perhaps, or some structure that could only have been made through a living entity. And so if you start reading into this area of study, they refer to these ancient cells as protocells. So you might see that term, protocells. And these are when I was first taking microbiology classes when I was an undergrad, they thought life first arose on Earth, like in the shallows of the oceans, like along the shoreline. That has since changed. They now think that it is deep in the ocean, and in particular around hydrothermal vents or seeps in the ocean. I don't know, has anyone taken the evolution class here? Does that ring a bell? Okay, is that what they're saying in that class too? So I just, I'll show you a slide that I just inserted because I just read an excerpt of a paper this morning where they're looking, and again, so it's just published in 2024, and again, they're saying the hydrothermal vents is where we see, or where they hypothesize that early life first arose. And so it's shifted. When I was an undergrad, it was the ocean shore. Now it's deep in the ocean. And the evidence keeps accumulating that it's deep in the ocean. And so, so far anyways, that's the most logical idea currently. So they often show things like this, where you're looking at the time scale in these big swaths of time laid out. And you can see with the yellow bar, they're saying all of that swath of time was microbial. And so I like this image because it's trying to point out that most of Earth's existence, it was microbial life. That was it. And then eventually you start to see other organisms to existence, speciation occurring. So you can see that the on the left here, you see the earliest microbial life, and then it works its way up. You can see, you're getting just like formation, and then I'll talk more about mechanizing when we get into cell structure. But you can see here that you're getting into, early eukaryote showing up. We'll talk about that as well. And so, they're like trying to hunt for fossils and some kind of existence. So these are images here are what they consider fossil evidence of microbes. And so maybe to your eye, because you're not trained in yet in lab on what microbes look like, under the microscope, but that, take my word for it, those do look like microbes. And so there's some fossil evidence, but we're talking about scanning these fossils, with a microscope. It's incredibly challenging and kind of needle in a haystack idea on how to find these. Come on, advance. Doesn't want to advance on me. Okay, let's see. This is another image with a little bit more of a chemistry of what might be happening. Put on some time scale again. And so at the bottom of this image, like first it was just chemicals floating around in those hydrothermal vent areas. They pose that the clay surfaces of the hydrothermal vents are good physical places for some of these reactions to take place. That clay allows for certain chemicals to easily commingle and start to polymerize. And so we also see molecules that the carbonate, sulfur, iron should have been there. And then they're saying, through these hydrothermal vents have the right conditions of clay surfaces and the right temperature, the right pressure, that you can get some of these chemicals and molecules reacting with each other to form more complex amino acids. And the paper that I just read this morning, they were looking at lipids that form the membrane of the cell. And so you can see here prebiotic chemistry. And then I think they teach this now in the current evolution class too at the macro level. When you think about evolution, you have to think about storing information in these cells, and they propose that it was actually RNA that came along first. It's simpler than DNA, and that it actually was a way for cells to store information. And then gradually over time, you can see the shift where now DNA shows up as the information storage molecule. So, by the way, do you remember what LUCA stands for? LUCA? Last universal common ancestor. And then you start seeing speciation off of that into eukaryotic, for example, cell type. So here you can see at the very top, speciation going on where you have bacteria, and then you see that word archaea. So I'll talk a little bit more about those domains. And then this inset picture, that's what an actual hydrothermal, that's a black smoker hydrothermal vent that you're looking at. So you have to go deep into the ocean, start thinking about the idea of these molecules interacting with each other under the right physical and chemical conditions to create more complex molecules that make up, organic organisms like us. But first it started off with just those ancient protocells. And so Here you're looking at a very simple chemical reaction on the left there, where they're showing some heat. That squiggle line usually means heat input, electrical discharge. And so they're saying, from these molecules floating around next to each other under the right conditions of temperature and pressure, you can get more complicated, more complex molecules. Glycine, which is an amino acid. All right. So Stanley Miller was the scientist at the time that one of the first earliest scientists to show that this can happen under the right conditions. You can get more complex molecules. So, okay, so it seems that hydrothermal vents could provide good conditions for polymers to form and more complex molecules to form from simpler ones. But in order to get a cell, you have to have a cell membrane of some sort. And so thinking about that idea, there's been a lot of evolutionary microbiologists that have looked at that too. And really though, when you, excuse me, if you have phospholipids, I mean, you can do this and just take a beaker of water. If you have some phospholipids, dump it into the beaker of water, and you'll see that it will rearrange into a phospholipid bilayer membrane configuration, because phospholipids have regions that like to interact with water and other regions that don't. And so you get this phospholipid bilayer form. So that can happen pretty spontaneously when you get the phospholipids made, through these chemical processes. So this was the article that was just published that I mentioned. And so they are posing that, yeah, this is doable in the hydrothermal vent area. Long chain fatty acids, so phospholipid bilayer membranes are long chain fatty acids, part of their composition. And they're saying, in the hydrothermal vent, you can have the right conditions for this to happen. And so they're continuing to build evidence on this idea that protocells were forming in the hydrothermal vents all the way to cellularity, getting those cells to form. And so again, looking at stromatolites is an excellent way to look for these ancient protocells. These are just different views and sizes of stromatolites. This stromatolite here that they've taken a sample of, and you can see the layering of the microbes landing. And so this one's about 3.5 billion years old. And then, believe it or not, there's actually stromatolites here in the UP. And so this is an example of one of them. And Chocolate Hills, located southeast of Ishpeming, Michigan. 2.2 to 2.4 billion years old. So we do have our own stromatolites here. Not as old as most of the oldest ones so far are showing up in Australia, along the coast of Australia. But we have our own here. Here's more very beautiful fossilized stromatolites. And so, taking samples of these and looking for fossils from here, are on the left are the microfossils, and then on the right, they're trying to show a comparison picture to that fossil of living microbes. And so if you just compare the left images to the right images in pairs like that, you can see that they do look like, current living microbes. And so hats off to the people who do this kind of research, because it is amazingly complicated and difficult. Here you're looking at a scanning electron microscope image. And again, since you've not been into lab yet to look under the microscope, but you can see, I think, if you just kind of squint and look hard, you can see that what they're pointing at there, it looks like a rod-shaped cell in this microfossil. And so that's what they're hunting for. They're taking, like those stromatolites, they'll take samples of them and then look at them under the scanning electron microscope and look for the typical shapes of microbes. And rod shape like that, very, very typical. Okay, so 3.5 billion years ago, that number I've seen, some people are now proposing a little bit older, like cells came out around 3.7, 3.8. But let's stick with 3.5 for right now because the evidence still needs to build some more. 3.5 seems to be a safe number to think about in terms of when did those protocells arise. But, getting a cell to be totally, I mean, if you think about it, there had to be just millions of iterations of, trying to get all those complicated molecules all together and then enclosed in the membrane to actually have a functioning cell. It's really kind of mind-boggling to think about. And then once you get a cell, well, that's not the end of the story, obviously, because then you have to think about, well, how does the cell do its thing in order to stay alive? And in order to do that, to stay alive, one of the things you have to do is be able to get your version of energy, which is ATP. And so that requires some biochemistry. You have to have a physiological pathway in order to generate. So evolutionary biologists have thought about, how could that have happened roughly 3.5 billion years ago, given those conditions of Earth? And there's no oxygen back then. So it couldn't have been through oxygen mechanisms. Like what you're doing right now, you're sucking in oxygen and you're creating a lot of ATP in your mitochondria with the help of oxygen. They didn't have that 3.5 billion years ago. So they had to figure out some other strategy through natural selection. And so they think about, well, what were some molecules present back then that could have played a role? Iron, you're seeing here, hydrogen could have been, and sulfur could have been some of the ones that played a role in getting the first biochemical reaction to generate some ATP. So let's look at this a little bit and dissect it. So what they're trying to show here is a membrane of a cell. So this is a protocell. And you're seeing a couple of things that are spanning the width of the membrane. You have a hydrogenase, okay, and then an ATPase. Now, you have a couple of things happening. The things above the membrane are what's happening outside the cell. And so what they're showing here are a couple of reactions. And in the box, that is ultraviolet light. They're saying if you can have some ultraviolet light energy in the area where there's some iron and some protons floating around, you can spontaneously, with the help of ultraviolet light, get this reaction occurring where the iron and the protons react. So Fe2 plus H plus, and you end up with hydrogen gas, H2, and then the leftover ferric form of iron. But it's the hydrogen gas that we want to zoom in on here. So that's a good thing because now look what's happening. That hydrogen gas gets produced and the cell says, I'll take that. Thank you very much. And so what you see happening here is the hydrogen is actually getting stripped of electrons. This hydrogenase is going to work on hydrogen and say, give me some of your electrons. I want those. And so that's what's happening here. This 2 electron is getting passed. So they're saying electrons get grabbed. Remember, each hydrogen has a proton and an electron, okay? So if you have 2 hydrogens, you can grab 2 electrons off of that. And so 2 electrons are getting grabbed by the hydrogenase and being brought into the cell. Well, that's important because those electrons are going to help complete that reaction going on inside the cell, which is water plus S to the 0 is elemental sulfur. So there's plenty of sulfur back then, they hypothesized, all right? So the cell has some sulfur, it's certainly got a lot of water in it. And with the help of those electrons getting stripped off of hydrogen gas, you run that reaction. Well, why should that matter? What's the big deal about that? Well, it's kind of a domino thing here going on. So you see this H2S is one of the products, and then you have a hydroxyl. Okay, so let's look at the H2S. That's hydrogen sulfide. If any of you had the joy of making that in chemistry class, it smells like rotten eggs. So hydrogen sulfide is a gas, smelly gas to our nose anyways, and it's a toxic thing. You don't, including a microbe, doesn't want too much of this. So that's why you see this hydrogen gas. It's like, yeah, I don't really want that. I am going to push that out of my cell. So they're getting rid of it. And that's a good thing. First, because it's helping keep the microbes safe, and it's feeding into this reaction that's going on outside the cell. where it says, I'm going to get rid of that hydrogen sulfide. And then if there's some iron sulfide there, those two can react. And then you get this FeS2 plus hydrogen gas. So there's more hydrogen gas being made. So you can get hydrogen gas being made spontaneously in the atmosphere, plus the microbe can contribute to making sure there's some hydrogen gas. If you can keep this going, it's kind of like this loop going around and around and around. There, get that in reaction, sulfide, and back out, keep that reaction going round and round and round. Okay. Now, on the face of it, it's like, well, so what? What's the, what's the payoff here? There needs to be a payoff for doing this, right? Okay, well, let's go back to that hydrogen gas. When you look at the hydrogen gas, there's something else going on here. So we said hydrogen gas is being used, the microbe saying, give me those electrons, and that brings those electrons. When you take electrons off of hydrogen, then you're left with the proton. Okay, so now you see that's the other product. Strip electrons off of H2, you're left with protons, 2 protons in this case. Now we're talking. This is a really good thing. I wish the artist had been a little bit more flamboyant with hydrogen, the H plus protons. He or she should have drawn many, many protons. So if you have this reaction happening over and over and over, where this keeps going round and round, you're going to get a buildup of protons, more and more protons, more and more protons. Okay. Well, then what that does is it creates a gradient of protons across that membrane. This is a really important concept. I am going to talk about this a lot in this slide. Right? Proton gradient. More protons outside the cell, less protons in the cell. By the laws of diffusion, those protons will want to diffuse across to even out the concentration, but they can't do that just through the membrane because they're charged. They can't diffuse it as a polar medium. It won't work. So they use the primitive ATPase to do that. So they're trying to show that these protons are coming through the ATPase. And as that is occurring, as those protons are flowing through the ATPase, it actually causes that ATPase, creates a torque force, actually. And these subunits down here of the ATPase actually rotate and allow for ADP, an inorganic phosphate, to physically react with each other, come into contact. And then you see the huge payoff. Once you get that reaction to go, then you make ATP. All right, so ATP, when you look at this whole thing going on, the whole goal here, the game that the microbe is playing, is to create as simple of a process as possible to get some ATP. And once they accomplish that, then, that's their chemical energy. We all function on ATP, including those protocells 3.5 billion years ago. So really, it's like the microbe is setting the stage. What can I do? How can I work things to set the stage to allow myself to get a proton gradient, which then will allow for ATP production? Questions on this? Does this ring any bells for those of you who've had some biochem maybe, or maybe BI-218, intro to cell and molecular? Okay. This is a theme I come back to a lot in this class. So if you're sitting there going, ooh, yeah, I haven't thought about this in like forever, start thinking about it now, please. Okay, start working through this. And so this concept of what ancient cells were doing, so evolutionary microbiologists are still pointing to this. A lot of them still think this was probably the first biochemical reaction that evolved. to allow for ATP synthesis. So I'd like you to be comfortable answering questions on this, on the exam coming up during week 3. It's coming up fast, okay, week 3, okay? Questions on that? Well, let's give it some study over the weekend, look at it a little bit, and then Monday we'll go through it one more time and I'll answer any questions you might have. But basically, it is, I like to think of it as games. The microbes are playing games, and there's certain rules of these games that they have to follow. And they're, you want to win the game. There's some goal. And in this case, the goal is ATP. And so stringing together some of these physiological pathways, this one actually, believe it or not, this is really simple. It's a pretty simple mechanism here. If you think about some of the other pathways that have evolved, like glycolysis and citric acid cycle, much more complicated. And, those are also used for ATP production. I like this depiction of the evolutionary timescale in the sense that it is giving this clear demarcation between what's happening in the absence of oxygen versus the presence of oxygen. And so if you can imagine these protocells, developing these physiological pathways, figuring out how to make ATP, figuring out how to take in nutrients and get those in the pathways and build their cells, reproduce themselves, make ATP, and then of course they start, the speciation is occurring. And so all of that's happening over, a couple billion years. And then you start to see the brown coloration down low versus the blue. So right in that demarcation shift from brown to blue, that's the great oxidation event is often how they refer to that time period. And so what's happening here is that the microbes, the ancient microbes, are busy doing their thing. Initially, in the absence of oxygen, there was no oxygen on the face of the earth, okay? And it's because of the microbes that we now have oxygen. That's who we have to thank for us allowing to come on board and exist on. And so here you see that you have different groups of microbes in that brown area showing up. And so we'll come back to these microbes. You probably are most familiar with the cyanobacteria. They're showing up late on that brown section there. And so I think some of you may know that cyanobacteria can do photosynthesis. And so they do photosynthesis very similar to how plants do it. So if you've taken plant phys, they're doing it very similarly, okay? And so they're producing oxygen. So that's why they're right on the line there, okay? So cyanobacteria are really who we have to thank for this kickoff to getting our atmosphere oxygenated. And you can see oxygenic photosynthesis is what they do. That just means oxygenic means the creation of oxygen, oxygenic. But if you look a little further down, you see anoxygenic photosynthesis. So I just wanted to point that out now, and I'll come back to it. So the so-called purple and green bacteria, they did photosynthesis first before cyanobacteria, but they still didn't have enough oxygen to really work with. And so they did an anoxygenic. Anytime you see A or AN in front of a word in science, that means without. So anoxygenic, without the creation of oxygen photosynthesis, right? So we'll learn a little bit about that. So there's photosynthesis out there that is different than how plants do it, for sure. And so then you get that great oxidation event and aerobic respiration, which is what we do. So we have to go through aerobic respiration to make ATP. We have to use oxygen in our mitochondria. So you can see, eventually the first eukaryotes with organelles arose, and then multicellular eukaryotes came out after that. Right, and then we're like, wait, we're a blip on this whole thing. We're just a blip. So microbes rule. They have been here 3.5 billion and onward, and we have them to thank. So microbes are everywhere. And, initially, when I was first taking microbiology, there were just two domains of life that they had, microbes classified into. And now they say, no, really, there's three. And it's because of Dr. Wolf that we now say, no, there's three domains of life. He was the one who really started to realize through his pioneering work where he said, why don't we take SSU, that stands for the small subunit of ribosomal RNA, why don't we use the small subunit of ribosomal RNA as a comparison across all living organisms, and let's see how it pans out. And so it's a good tool in order to look at phylogeny and relatedness. And when he started doing this, he realized that the so-called archaea type of ancient, well, they're current ones too, microbes, just microbes, the archaea type microbe, we used to lump them into the bacteria, and now they're like, yep, they're different. And so now we have three domains. I need you to know what the three domains are: bacteria, archaea, and eukarya. Of course, we belong into the eukarya. But the bacteria and archaea, those are microbial, and they're prokaryotic. So we're eukaryotic, they're prokaryotic, right? And then you can see the last common ancestor on there as well, and then the deviation out, speciation. And so when they started doing this work with, more recent technology, you can see that, well, especially if you talk to evolutionary microbiologists, the second point is pretty well accepted. I don't think it's as well accepted out when you talk about evolution to people who aren't microbiologists, but a major engine of evolution is symbiosis. And I think you get taught that a little bit in the evolution class, and I'll mention a little bit about that here in a second. Well, right now, I guess. So when we think about this shift of, okay, 3.5 billion years ago, ancient cells came about. They were prokaryotic, no organelles, no nucleus. And then you have to make this shift into a more complex cell, the eukaryotic cell that has all these organelles, and they're bigger in general. How do you make that shift? What happened to allow for that huge jump in evolution? And even in typical evolution classes, I think this is commonly accepted as the most plausible idea, is that you had merging of cells billions of years ago that allowed for the eukaryotic cell type to evolve. And so this idea of endosymbiosis, okay? And so, no matter where the microbe is, whether it's inside your intestines or on the cliff face in Antarctica, you have this idea of symbiosis going on. And it, when you get down into the protocells, if you shrink down into that, if you think about, let's say there's two protocells swimming around those hydrothermal vent areas, what if a slightly larger one engulfed a smaller one and was trying to use that smaller one for nutrition? That's probably how it all started, perhaps. And then what happens if that happens, where the smaller one gets engulfed and then the larger one doesn't kill off that smaller spot and instead just lets it live inside? And that's because there's some kind of benefit, there's a payoff, all right? And so it can be a win-win situation. And so when they started thinking about these ideas, this idea of a cell within a cell, this endosymbiosis idea could also be the mechanism that allowed for going from prokaryotic to eukaryotic billions of years ago. And so that's the endosymbiosis theory that has been proposed, and it's typically talked about in every intro biology textbook. It was proposed here in America by Dr. Lynn Margulis. Actually, the endosymbiosis theory that she was working on this back in the 1950s and 60s, I think. And that was during the Cold War, when the United States and Russia were very much at odds. And we now know that Russian scientists were also looking at this. And so it's not just her, but she's largely credited with it because she had a lot of data to support this. So she actually did this for her PhD thesis. This was her PhD thesis in the 60s, where women scientists weren't common. And so when she went to defend her graduate work, it was not well received. And she actually was criticized and told, why don't you just, be a wife and mother? And it was, I met her a couple of times, and she came up to Michigan Tech when I was a grad student. And it was really, really interesting to get a chance to speak with her. Fascinating wife and an amazing scientist. And so, but, time, and more data supported what she was talking about in her PhD work. And more data and evidence has been proposed and shown to support this endosymbiosis theory. So basically, we're thinking about this idea that a cell within a cell could occur. And so your mitochondria, the mitochondria in our cells, that's the idea of a cell within a cell. Mitochondria were once free-living ancient microbes. And then a bigger cell came along and engulfed it and said, you know what, I like that you can use oxygen. That's actually quite handy for me, because back then when oxygen first started to accumulate, it was poisonous because microbes didn't know how to deal with it. They're like, I have no mechanism for this. And so I could wipe them out. Well, if you could swallow, so to speak, engulf a cell that knows how to use oxygen to make ATP, wouldn't you keep it? That would be a winner. That's a keeper. All right. So they think that's how mitochondria arose in the eukaryotic cell, is that they were able to use oxygen to make ATP. Perhaps they got engulfed by a bigger cell that couldn't do that. And then that bigger cell is like, yeah, I like this. I can now go into areas where oxygen is prevalent and I don't get poisoned. I stay alive and I get a ton of ATP, because using oxygen is a great way to make ATP. So that was one of the things that she proposed in her PhD thesis. And she had, some evidence to support this idea. And then what if you engulfed a smaller cell into you that could use sunlight to make sugars and ATP? I mean, how awesome is that? Wouldn't you love to just go to the beach and lay out for 15 minutes and you'd be fed for the entire day? It's like, I don't have to cook any meals. I just have to get some sunshine. And so this idea of the chloroplast was once free living and then was incorporated into a larger cell and then kept alive and modified into what we now call the chloroplast. And so very, very, when this was first proposed, you can understand perhaps why it was met with a lot of criticism and disbelief. It's a kind of a out there idea. But once you start thinking about it, it's like, it really does make sense that this strategy would be put into play all those years ago to allow for these microbes to survive and adapt quite nicely. So she proposed some evidence. I'll list out a little bit of it. With the chloroplast and the mitochondria, they both have DNA. So they have their own DNA, which is what organisms need for information storage. So why would chloroplast and mitochondria have DNA in our cells unless perhaps they were once free living and they needed that DNA for their solitary existence? Ribosomes are similar. When you peer inside chloroplast and mitochondria, they have ribosomes, and they are snow-like bacterial ribosomes. In fact, if you expose chloroplast and mitochondrial ribosomes to antibiotics that can target bacterial ribosomes, the ribosomes fall apart. It works. So that's another piece of evidence. The cell membrane, the cell membrane of the chloroplast and the mitochondria are very similar to bacterial cell membranes. And just physically, they're the same size. Bacteria and chloroplasts and mitochondria are about the same size as typical bacteria. So there's other evidence. I'm not going to keep going, but the evidence has grown in this idea. So endosymbiosis theory, is this ringing bells? Okay, it should. I'm guessing in your intro biology and in the evolution class. So here's an example of a cell within a cell. So here you have the big cell is algae. Or protozoa, sorry, big protozoa. And then chlorella. And so you see the protozoa is jam-packed with chlorella. And you can see the green kind of gives you the hint that it's doing photosynthesis. So this protozoa is like, sure, I'll take in some chlorella because then they can do photosynthesis for me. I don't have to do it. And I get sugars, ATP, that they'll share with me. And so this is an example. Here's another one where this Tatina, a protozoa that has a lot of green algae inside of it. And it's interesting when the one on the left is like the adult cell that then goes through cell division. And you can see that one of the progeny cells is green. So one of the progeny cells gets all the algae. The other one is left with nothing. And so this one has to go swim and collect its own. So one is clearly favored over the other. One kid's favored over the other. So anyway, so these are just some examples of this idea. And so that's Dr. Margulis. And so she actually, she was married for a while to Carl Sagan. Do you know Carl's that name? He was a famous astrophysicist. You ever see the movie Contact with Matthew McConaughey? He wrote the book that movie's based on. Anyways, he's a well-renowned astrophysicist. So he was married to Lynn Margulis. Can you imagine their dinner conversations? They had a son, Dorian, who's a scientist. And so Lynn and Dorian wrote a lot of books, scientific books that are actually really easy reads, not a bunch of jargon. It's not that technical. Very, very interesting. So one of the quotes from her on this idea of symbiosis, this merger between microbe and animal cell or microbe and plant cell, is really different from the ancestral cell that lacks the microbe. Analogous to improvements in computer technology, instead of starting from scratch to make all the modules again, the symbiosis idea is an interfacing of pre-existing modules. Mergers result in the emergence of new and more complex beings. I doubt new species form just from random mutation. So random mutation certainly is a thing that drives natural selection. But if you talk to an evolutionary microbiologist, they will say, absolutely, this idea of symbiosis is a major way that we see evolution and speciation occurring. And so, the endosymbiosis theory is kind of what started it all. They're saying this is still a thing. It's very interesting reading. And so here, this was taken from a New York Times article, a summary of a scientific paper. I just love the image here where they're showing a phylogenetic tree of life. And, this little thing down at the bottom, that's us. That's the eukaryotic domain, eukarya. But look at everything else. That's all microbial, prokaryotic. So this is a nice visual kind of hit you in the eye idea that the vast majority of life out there, the vast majority of species out there are prokaryotic microbes. So I think that's a really cool depiction of the phylogenetic tree. So I often think about this quote, in biology, nothing makes sense except in the light of evolution. So as you are thinking about what's going on with microbes in this class, I ask you to keep this in mind. So, for example, when we get into the physiology section, it's like, would they do this? Why do they do this versus doing that? Well, let's think about this idea. It's this idea of, well, where is the microbe located? What kind of resources do they have around them? How are they going to make a game out of that to meet their goals in the game? And so again, thinking about the evolutionary process really, I think, does help kind of figure out why microbes do what they do. And so hopefully, as we work through the class, if you just try to keep that in the front of your head, you'll start to see some interesting aha moments. So, and I couldn't resist this one. So, anyways, I think we'll stop there. The next topic, I'm still getting into evolution, but we'll do that starting on Monday. Okay, so try to get your iClicker squared away. Please look over that ancient physiological pathway that we were looking at for making ATP, and we'll review that on Monday.